Hello everyone, today we will be solving Cambridge International AS and A-Level Biology Paper 5 Planning, Analysis and Evaluation, May June 2021, Paper 5-3. First question. When bananas ripen, they turn brown in color. This is due to the formation of a brown product called melanin. The enzyme catechol oxidase acts on its substrate catechol, leading to the formation of a brown product. Melanin shown in figure 1.1. So catechol, pale pink substrate, all right, and then catechol oxidase oxidizes it to melanin, so we can get the brown product. As melanin is produced, the color of the reaction mixture changes to brown. The intensity of the brown color produced is proportional to the concentration of melanin. The more active the enzyme, the more intense the brown color. A colorimeter is used to measure the absorbance of the reaction mixture. Absorbance is a measure of light absorbed by a colored solution with the reaction shown in figure 1.1. The more intense the brown color, the higher the absorbance. Catechol oxidase can be extracted from bananas. Some students were asked to extract catechol oxidase by grinding a slice of banana with sand and water using a mortar and pestle. The resulting mixture was then filtered through a piece of cloth. Suggest a reason for grinding with sand. So basically, grinding with sand will allow basically to that banana to release those, break open those cells and release those enzymes. All right. And then filtering with a cloth will basically allow sand and debris to be removed so that we only get the enzyme. Part B. The students investigated the effect of substrate concentration on the enzyme catalyzed reaction shown in figure 1.1. The students were provided with the extracted catechol oxidase enzyme solution prepared in step A, which was kept cold until needed. A stock solution of 1% catechol solution made up in a buffered solution of pH 7, a buffer solution of pH 7. The procedure used by the student is outlined in steps 1 to 3. The extracted catechol oxidase enzyme solution was mixed with 1% catechol solution. After 2 minutes, a colorimeter was used to measure the absorbance of this mixture. Step 1 and step 2 were repeated using different concentrations of catechol solution. Suggest why the students used a colorimeter to measure the absorbance rather than judging the intensity of the color by eye. You see, often judging the color by eye, it is not quantitative, it is rather qualitative. When using a colorimeter, the result is quantitative. Part 2. Identify the independent variable and the dependent variable in this investigation. Independent variable. So basically, when talking about independent variable, we have to understand this is the particular reagent that we are either changing concentrations and that we have control over, and we are going to change this particular parameter. So in this case, we can see we are provided with the stock solutions of 1% catechol solution which is made in a buffered pH of 7. All right, and we want to investigate the effect of substrate concentration, which is in this case it is catechol. So the answer will be the concentration. Independent variable will be concentration of catechol or the substrate. Then we'll have to think of the dependent variable. So what is the dependent variable? A dependent variable is basically the absorbance on a colorimeter, because the colorimeter is going to absorb light depending on how much yellow color is formed. The students use the absorbance values at 2 minutes as the initial rates of reaction. Figure 1.2 shows the results. So percentage concentration of catechol as it increases, initial rate of reaction increases. We can see after a certain concentration, it plateaus. Part C. The students decided to investigate the effect of inhibitors on the reaction shown in figure 1.1. The student planned to add an inhibitor Y to the reaction mixture containing different concentrations of catechol solution. Describe a method that the student could use to collect the data needed to test the effect of inhibitor Y in a reaction mixture containing different concentrations of catechol solution. The description of your method should be set out in a logical way and be detailed enough for another person to follow. You should not repeat the details from A describing how the extract of catechol oxidase is prepared. Okay, so in a question like this, all right, we are given a stock solution, so definitely we must prepare several several different concentrations of that particular catechol. Okay, and then because we're using an enzyme, and basically the enzyme, it is already mentioned that it has to be prepared, which has to be kept cold. All right, the enzyme has to be prepared beforehand and it has to be kept cold until it is needed. So we must also have a method to keep that particular enzyme solution cold before the experiment. All right, after that we need to have certain parameters of those experiment, like enzyme is enzyme reactions are affected by pH, enzyme reactions are affected by temperatures. All right, so we must standardize those things, we must keep them constant, and also we must keep the volume constant. We must keep the volume of the substrate, the volume of the enzyme, and also the volume of the inhibitor constant throughout the experiment. And by keeping all of these things constant, we will repeat the experiment at different concentrations of catechol and measure the intensity of the color that it produces. And we are going to measure the intensity of that particular color at a set amount of time, because we want the reaction to continue. So like 1 minute or 2 minutes would be a very good, or like 5 minutes could be a very good amount of time that we can allow that particular reaction to continue. And then what we can say is we can talk about repeating on the same concentration of catechol several times so that we can get average. And then what we can do is that we can also repeat the experiment in order to understand that whether that particular inhibitor Y is having an effect. We can repeat the experiment without the inhibitor Y, and instead we can replace it with water. This particular experiment is a relatively low risk experiment. However, we are using a particular enzyme called catechol oxidase. Enzymes are known for their irritations. So obviously wearing gloves and the face mask is very important to avoid coming in contact with the enzyme. So the question said that we will have to describe a method that the student could use to collect the data needed to test the effect of inhibitor Y in reaction mixture containing different concentrations of catechol solution. So our description should be in a logical manner. So what we are going to do, because we are provided with a catechol stock solution, as you can see, we're provided with a 1% catechol stock solution. So we're gonna do a proportional dilution to get few other concentrations, because we're gonna see the effect of the concentration of catechol on the rate of reaction. All right, and then also the inhibitor. So the student can prepare at least 5 concentrations of the catechol. For example, 1%, 0.8%, 0.6%, 0.4%, 0.2, and 0.0%, which is basically distilled water. And the 0.0% can also act as a control. The enzyme can be kept at cold by using cold water bath. Because the enzyme, it is mentioned in the very beginning that it has to be cold until needed. The catechol oxidase must be cold until needed. So that's why once collected, the enzyme should be kept in a low temperature. The enzyme and the substrate should be equilibrated to room temperature before mixing. So let's say if we're doing the experiment at room temperature, definitely it has to be equilibrated up to room temperature. If we're doing that particular experiment at a higher temperature, of course we should do the equilibration up to that temperature. Now all reactions must be carried out at the same temperature. All right, and a water bath can be used to ensure constant temperature. For all experiments, same volume of inhibitor Y is used. All right, and then same volume of catechol and same volume of catechol oxidase should also be used. For all experiment, the colorimeter reading should be taken at 5-minute mark. For each concentration of catechol, at least 3 replicates and average should be calculated. The experiment should also be repeated without the inhibitor Y and use distilled water instead for comparisons. This is a relatively low risk experiment. However, some people can be allergic. However, we know that some people are allergic banana, so gloves should be worn. Figure 1.3 shows the results when no inhibitor Y was added. Initial rate of reaction, percentage concentration of catechol, no inhibitor Y. The student suggested that inhibitor Y was acting as a non-competitive inhibitor. On figure 1.3, sketch the curve expected if inhibitor Y was acting as a non-competitive inhibitor. Vmax is the maximum initial rate of the reaction of enzyme. Okay, so basically if we are asked to do a graph for non-competitive inhibitor, we must understand that it must plateau below that of the original line. So we'll start from the same origin, but we're going to plateau at a lower level. We will label it inhibitor Y.
Present.
All right. Now the question says, the Michaelis-Menten constant Km is the substrate concentration at which the initial rate of the reaction is half its maximum value. Draw on figure 1.3 the position of Vmax and Km of the enzyme when no inhibitor Y was present. Okay. So when no inhibitor Y was present, we can see it's 2.3. All right. So 1.8. So half of 1.8 will be 0.9. So this is 0.9, and then we're gonna go down. So that is basically the Km. So this is the Km, this is the half Vmax, and this is the Vmax. Part 4. Use your graph to describe the effect of the addition of inhibitor Y on the Km of this enzyme. All right. So because use your graph to describe the effect of the addition of the inhibitor on the Km of this enzyme. All right, so I think we can remodify the graph a little bit more so that we can match. So this is the half Vmax with the inhibitor present, and we can see the same Km. The reaction with non-competitive inhibitor has the same Km value with no inhibitor, which indicates that it is a non-competitive inhibitor. Part D. Hypertension is a condition where people have long-term high blood pressure. The two main types of drug used to lower blood pressure in people with hypertension is ACE inhibitors and diuretics. Scientists investigated the effectiveness of the drug when used separately and in combination. 505 people with hypertension were split into four groups as shown in table 1.1. Group 1 placebo, no active drug given. Group 2 ACE inhibitor only, diuretics only, ACE inhibitor and diuretics. The results are shown in figure 1.4. Mean reduction in blood pressure / kilopascal. Group 1, group 2, group 3, group 4. Okay. So, mean reduction in blood pressure, we can see the ACE inhibitor and diuretic group got the, highest mean reduction in blood pressure. Now, the question says, the scientists analyzed the data and concluded that combination of ACE inhibitor and diuretic should be used to lower blood pressure in the treatment of hypertension. With reference to the table in 1.1 and figure 1.4, discuss the conclusion that a combination of an ACE inhibitor and diuretic should be used to lower blood pressure in the treatment of hypertension. So we can see that both, the combination of the drugs gives a better reduction in mean blood pressure. So it's more effective. All right. So, and there are no error bars. So the error bars does not basically overlap. All right. However, there is no data on long-term results. Since the question says discuss, we can say both for advantages and disadvantages. So the question was, the scientists analyzed the data and concluded the combination of ACE inhibitor and diuretic should be used to lower blood pressure in the treatment of hypertension. The question says, with reference to table 1.1 and figure 1.4, discuss the conclusion that combination of an ACE inhibitor and diuretic should be used to lower blood pressure in the treatment of hypertension. In questions like this, we must look into the data. Once we get a particular relationship with the particular data that we are looking for, then we must also make sure that there are no error bars that are overlapping. We must also make sure that there is no statistical test that has been done between the groups to compare. And we must mention them, because based on what prediction can you think that both of these drugs can be used? Of course, we can also see that there are no data on side effects. There are no data on the age and gender of person, those who are being treated. All of this would be important to actually conclude that both ACE inhibitor and diuretic are actually good for hypertension. Question number 2. Inheritance of wing shape and eye color in fruit fly, Drosophila melanogaster, is controlled by two genes. The gene capital N and small n controls wing shape. Capital N for wrinkled wings is dominant to the small n for normal wings. Gene capital E controls the eye color, and allele capital E for rosy eyes in dominant to allele for red eyes. A biologist predicted that if genes are on different chromosomes, the ratio of the phenotypes of the F2 generation would be 9:3:3:1. Now, the biologist carried out a breeding experiment. Homozygous dominant fruit flies with wrinkled wings and rosy eyes were crossed with homozygous recessive fruit flies with normal wings and red eyes. All the F1 fruit flies had wrinkled wings and rosy eyes. The F1 fruit flies were crossed with each other. Alright, and table 2.1 shows the result of the F2 generation. we can see wrinkled wings and rosy eyes parental frequency in higher quantity than wrinkled wings and red eyes in lower quantity. Now the question says the chi-square test was used to analyze the data in table 2.1. State one reason why chi-square test was used. So the reason why we always use a chi-square test is we want to compare the observed and the expected data and the ratio of the phenotypes. To do a chi-square test, obviously the data must be categoric and the data must be discontinuous. So one reason why the chi-square test was used. Now the question says state the null hypothesis that the biologist would use for this test. Always we must the null hypothesis always says about one thing: there is no significant difference between the observed and the expected result. Part 3. Complete table 2.2 and calculate the value of chi-square for the results of the F2 generation. The equation for the calculation of chi-square is chi-square is equals to sum of observed minus expected whole square divided by expected. So we have wrinkled wings, rosy eyes, and we are working in a ratio of 9:3:3:1. So with that ratio in mind, the expected should be, now our total is 64. When 64 is divided by 16, all right, the answer is 4. So for 9 ratio it will be 36. Observed minus expected will be, observed is 44 minus expected, which is 36, divided by expected, which is 36, and it has to be whole squared. So the answer comes out to be 1.78. 12, 8.33, 12 and 8.33 again, and then 4, which is 36. Now our chi-square adds up to 54.44. Now the table 2.3 shows some critical values of chi-square at different probability. All right, degree of freedom. Now obviously our degree of freedom, because we have 4 sets of data, so our degree of freedom will be 4 minus 1, which is equals to 3. The degree of freedom is 3. And the exam probability is 0.05. When put together, is going to have a value of 7.82. State why the student used the value of 7.82 in this investigation. So, first of all, our categorical data, n minus 1, and probability. Since we have 4 classes of data, when 4 classes of data n minus 1, so it will be 3, and the probability in exam probability is 0.05. So that's how we get the value. Now the question says use your calculated value of chi-square to explain whether the null hypothesis should be accepted or rejected. Definitely the null hypothesis is rejected. And the question also says suggest a conclusion that the biologist could make about the inheritance of the genes controlling wing shape and eye color in fruit flies. So our, first of all, since we have rejected the null hypothesis, we're going to say there is a significant difference between the observed and the expected result. All right. And then we're also going to say that the genes are in the same chromosome. All right, so they're showing autosomal linkage, and they do not assort independently, which is why we did not get 9:3:1. So guys, that's all for today's video. Thank you for joining the video. And if you want videos like this, please subscribe to the channel and basically share the video with your friends. Subscribe to the channel and basically help the channel grow. All right, and best of luck with your exams. Bye bye.